Hell.
Hey there, welcome to our video. Today, we'll be discussing a groundbreaking study published in Nature Metabolism that explores the role of EphB4 in regulating insulin sensitivity and glucose metabolism. So, grab a snack, get comfortable, and let's get started. Abstract. In this study, the researchers investigated the impact of EphB4 on insulin signaling and glucose metabolism in mice. They used a variety of experimental techniques, including real-time PCR, Western blot analysis, and immunoprecipitation assays, to assess the effects of EphB4 inhibition on insulin sensitivity and glucose homeostasis. The results of their study revealed that targeting EphB4 improved insulin sensitivity and glucose metabolism in mice, highlighting the potential therapeutic implications of EphB4 modulation in the treatment of metabolic disorders. Insulin signaling is essential for glucose metabolism, and insulin decreases insulin receptor, InsR, levels in a dose-dependent and time-dependent manner. However, the regulatory mechanisms of InsR reduction upon insulin stimulation remain poorly understood. Here, we show that EphB4, a tyrosine kinase receptor that modulates cell adhesion and migration, can bind directly to InsR, and this interaction is markedly enhanced by insulin. Due to the adaptor protein 2, AP2, complex binding motif in EphB4, the interaction of EphB4 and InsR facilitates clathrin-mediated InsR endocytosis and degradation in lysosomes. Hepatic overexpression of EphB4 decreases InsR and increases hepatic and systemic insulin resistance in chow-fed mice, whereas genetic or pharmacological inhibition of EphB4 improves insulin resistance and glucose intolerance in obese mice. These observations elucidate a role for EphB4 in insulin signaling, suggesting that EphB4 might represent a therapeutic target for the treatment of insulin resistance and type 2 diabetes. Introduction. Insulin resistance and impaired glucose metabolism are key features of metabolic disorders such as type 2 diabetes and obesity. Previous research has implicated the EphB4 receptor in the regulation of insulin signaling and glucose homeostasis. However, the specific mechanisms underlying EphB4's role in metabolic regulation remain poorly understood. In this study, the researchers aimed to elucidate the functional significance of EphB4 in the context of insulin resistance and glucose metabolism. Insulin, a polypeptide hormone mainly secreted by beta cells in the islets of Langerhans of the pancreas, plays important roles in glucose homeostasis, cell growth, survival, proliferation, and metabolism. Insulin binds to the insulin receptor, InsR, and activates insulin signaling in downstream pathways, including the PI3K-AKT, mTOR, and RAF1-MEKERK1/2 pathways, to mediate its metabolic effect. Impaired insulin action, termed insulin resistance, is accompanied by obesity and leads to the development of type 2 diabetes and metabolic syndrome. Many in vivo studies have shown that insulin resistance is accompanied by hyperinsulinemia. Moreover, hyperinsulinemia, especially in the basal state, has been shown to sustain, expand, or initiate insulin resistance. For example, transgenic mice showing only a twofold increase in basal insulin concentration are characterized by severe insulin resistance. Furthermore, several in vivo studies in which insulin was administered to increase basal hyperinsulinemia to a similar level to that observed in insulin-resistant states have confirmed that the basal insulin level may lead to insulin resistance. Some studies have shown that continuous exposure to high levels of insulin reduces InsR abundance in lymphocytes, adipocytes, and hepatocytes. This supports the idea that insulin itself perpetuates insulin resistance to protect cells from constitutive pathway activation. At present, it is not known whether it is a physiological event of insulin on its receptor. Based on these findings, we hypothesize that mechanisms must have evolved to desensitize InsR signaling in insulin target cells. Materials and methods. The researchers utilized a variety of experimental approaches to investigate the role of EphB4 in metabolic regulation. They performed real-time PCR analysis to assess gene expression levels, conducted Western blot analysis to examine protein expression and phosphorylation, and utilized immunoprecipitation assays to study protein-protein interactions. Additionally, they employed pharmacological inhibitors and genetic manipulation techniques to modulate EphB4 activity in mouse models of metabolic dysfunction. Results. The results of the study demonstrated that targeting EphB4 improved insulin sensitivity and glucose metabolism in mice. Specifically, EphB4 inhibition led to enhanced insulin signaling, reduced hepatic glucose production, and improved glucose tolerance in mouse models of insulin resistance and obesity. These findings suggest that EphB4 plays a critical role in the regulation of insulin sensitivity and glucose homeostasis, highlighting its potential as a therapeutic target for metabolic disorders. Liver EphB4 knockout attenuates insulin resistance in mice. To investigate whether EphB4 is necessary for insulin-induced InsR degradation, we designed human EphB4-specific short hairpin RNAs, shRNAs, and conducted stable knockdown of HepG2 cells. In biotin-labeled cell surface protein assay, insulin-induced cell surface InsR reduction, whereas EphB4 knockdown almost completely rescued this effect. These results strongly show that EphB4 is critical for insulin-induced hepatic InsR degradation. Discussion. The findings of this study shed light on the functional significance of EphB4 in the context of metabolic regulation. By elucidating the effects of EphB4 inhibition on insulin sensitivity and glucose metabolism, the researchers have provided valuable insights into the potential therapeutic implications of targeting EphB4 in the treatment of metabolic disorders. These findings open up new avenues for further research into the development of novel therapeutic strategies for improving insulin sensitivity and glucose metabolism in individuals with metabolic dysfunction. In conclusion, the study's findings underscore the importance of EphB4 in the regulation of insulin sensitivity and glucose metabolism, and highlight its potential as a promising target for the development of novel therapeutic interventions for metabolic disorders. This research paves the way for future studies aimed at exploring the therapeutic potential of EphB4 modulation in the treatment of insulin resistance and type 2 diabetes.
PharmaPal.